Yes, I know. Thank you.
But remember,
it's fine if you do it wrong. Usually, I'm impressed you don't even have notes.
Good morning, everyone. Welcome to the seminar series in Ecology and Evolution of Montpellier. Welcome to people in the room and welcome to people online. For the people online, you can ask your questions by the Q&A tab that is at the bottom of your screen. We will activate the microphones in the end so that you can interact with the speaker. And we will alternate questions in the room and online so that everyone is able to ask their questions. You can use the chat if you have any technical issues, so don't hesitate to tell us if you don't hear properly or if any other problems occur. And I think with this, Giacomo, you can introduce us.
Yeah. Okay, good morning, everyone. My pleasure to introduce today's speaker at SEM, Yuko Ulrich. So Yuko is interested in social behavior, plasticity of social behavior, and also how disease may affect social behavior, but also the other way around. And so Yuko obtained her PhD in 2012 at ETH with Cornish Country Temple. She then moved to the US, Rockefeller University, where she started working with its ant system. And then she went back to Switzerland, Lausanne, starting her own research group. And then with the EFC, she moved to Zurich, and she's now at Max Planck Institute in Jena, keeping working on with this ant system. And so, without further ado, stage yours. And thanks again for being here. The seminar.
Thank you. Thanks, Giacomo, for the nice introduction and for the invitation to speak here. So it's really a pleasure to be here in person, especially after 2 years of a pandemic. And it's particularly nice to be in person in Montpellier. I've heard a lot about this place, but I've actually never been here in person before. So as Giacomo said, we are interested in behavior and disease in clonal ant societies. And to start with a very general introduction, I think me and my group, we are generally interested in how and why organisms live in groups, in the social behavior that they display when they do, and in some of the consequences of this social lifestyle. And of course, group living is a pretty widespread and also quite successful strategy, which can take many different forms in nature, from loose aggregations of pretty similar looking individuals in flocks of birds or swarms of locusts, through transient forms of multicellularity in some microorganisms, like those cellular slime molds, all the way to the really sophisticated groups of highly specialized individuals that fully depend on each other for survival, in some insects, some colonial marine organisms, and perhaps arguably also in humans. And a common feature of all those social groups, and one that I find particularly interesting, is that those groups are typically more than the sum of their parts, or in other words, they display phenotypes that no individual on its own can display. And for example, sociality is really at the source of some pretty remarkable innovation, like sophisticated communication systems between the different group members, or the ability to colonize new habitats or to exploit new types of resources. It also allows for division of labor and cooperation between the different group members, both of which are predicted to increase group level efficiency. And all of these, all those advantages are thought to have helped some social organisms like ants or humans to dominate many ecosystems. Now, of course, group living doesn't only have benefits. There can be various forms of conflict between the different group members, for example, over who gets to reproduce and who doesn't. And the need to feed many individuals in one place can lead to local resource depletion. But the one cost of social life that we have all been very aware of in the last 2 years is the increased risk of disease outbreaks that is associated with living at high density and engaging in frequent social interactions, considering each of those social interactions is also an opportunity for disease transmission. So the research of my group really focuses on the interplay between two of those aspects, division of labor and more generally social behavior on one hand, and disease dynamics on the other. And we studied those questions in ants. I have a few pop-ups. Okay, they are gone. All right, and yeah, we studied those questions in ants. As you probably know, ants are pretty extreme social organisms. They can live in colonies that can contain up to several million individuals, but that are integrated to such an extent that they often seem to function as a single unitary organism. And division of labor is really one of the key traits of their biology, and it can also take some pretty extreme forms. For example, between this large leaf-cutting ant queen, who is highly specialized in reproduction, and her many tiny and often completely sterile workers who take care of colony maintenance. Now, of course, maintaining an ant colony includes many different tasks, so there's typically also division of labor between the workers. For example, between this large worker who cuts the leaves and brings them back to the nest, and the smaller worker who rides on the leaves to protect her sister against parasitic flies. And although it's pretty clear that division of labor is central and perhaps even necessary to the functioning of social groups, the question of how it emerges in the first place and how it is regulated are still actively debated in the field. And then because ants are particularly social, you might expect them to be particularly vulnerable to infectious diseases. So in this picture here at the center, you see the density at which ants typically live, which is pretty much on top of each other. And from this, it's probably easy for you to imagine how a single infected individual might rapidly contaminate your entire colony. And so here, one important challenge is to understand whether some social groups are more resistant to disease than others, and if so, why. And one question that we are particularly interested in is to understand whether social organization itself can reduce disease risk. And this is because although sociality in itself is often thought of as increasing the risk of transmission, it actually also provides new ways to fight disease. And this is very obvious in ants, who have evolved a really wide range of collective and often behavioral defenses against pathogens. So in this image, for example, you see 3 ants that are grooming a 4th ant, the one with a red dot on her abdomen. And this is because this 4th ant has been exposed to pathogenic fungal spores. And this behavior effectively removes the spore from her body surface and will dramatically increase the likelihood that she survives. And at the level of the entire ant colony, the typical pattern of interactions between the different group members, represented here by the structure of the interaction network, has also been proposed to prevent disease transmission, with ants belonging to different behavioral groups having reduced interactions. So this is shown here by the fact that the foragers in red interact much more often with other foragers than they interact with nurses in blue. And they almost never directly interact with the queen, which is this dark blue node here, who again is the sole reproductive individual in the group. So those are just examples of strategies that are really targeted at protecting the entire group rather than the individual. So in principle, ants are pretty appealing systems, I think, to study both sets of questions, because they are naturally highly social and can also fairly easily be used in experiments compared to, say, vertebrates. But in practice, not all ants are easy to study, especially in the lab. So first, in most ant species, including all the images that I've shown you up to this point, the colonies are inherently so large and so complex that it can be difficult to keep track of colony composition and even harder to control it with any precision. And then there's also often very limited behavioral flexibility at the individual level. So in a typical ant colony, for example, the queen never forages and the workers never reproduce, no matter what you do. So to study those questions, I had long wanted to find a system that provided a bit more flexibility and also more control. And the system we work on in my group is called the clonal radar ant, and it's a species whose unusual biology naturally solves a few of those issues. So first, the native range of this species is in South Asia. It's shown in green in this map. And, but it has also been introduced, probably accidentally by humans, to various tropical and subtropical parts of the globe, including many islands in the Pacific. It's a doryline and therefore a relative of the more famous army ants. And as many army ants, it's a predator of other ant species. Like all ants, the species lives in groups in which individuals work together to take care of their young or to collect food. And they also display collective behavioral defenses against disease, like mutual grooming that I mentioned earlier. But now, in contrast to most ants, this is a species in which all individuals can take on any role. So unlike other ants, the clonal red ant actually has no queens. Instead, colonies consist of workers that all reproduce. Then, while other ants mostly use normal sexual reproduction, the clonal red ant reproduces exclusively asexually, meaning that all individuals within a colony are genetically nearly identical. And finally, reproduction is fully synchronized, with all colony members laying eggs on the same day, roughly once a month. And this means that all adults naturally emerge in discrete age cohort. So this is, of course, kind of interesting in its own right, but it's also very convenient for us because it provides us with the ability to precisely control and replicate not only individual age and genotype, but also several important aspects of colony composition, including colony size, genetic and demographic structure. So this makes it a system that is naturally more modular, can be propagated in the lab indefinitely, and allows to perform large-scale experiments. Okay, so today, in the first part of this talk, I'll describe some of my recent work on the emergence and regulation of division of labor in this particular system. And then I'll describe some of the current and also some of the planned future work of my group, which builds on those early results to try and understand whether social organization can reduce disease susceptibility. So the work on division of labor was part of my postdoc in the lab of Daniel Clonal at the Rockefeller University in New York, and was done in collaboration with other members of that lab, as well as with a team of theoreticians in the lab of Corina Tanita at Princeton University. So very broadly speaking, division of labor is simply the fact that different individuals in a group perform different tasks. And in ants, as in most other animals, there are many different factors that affect individual task performance. So for example, individual traits like genotype, age, or morphology often correlate with what an individual ant does. But because ants live in groups, what an individual does often also depends on who else is in the group and what those other individuals are doing. And that means that group traits like colony size or genetic structure are also expected to affect individual task performance. In nature, of course, all of those effects typically play out simultaneously and are difficult to disentangle from one another. So in this work, we really use the advantages of the clonal variant to try and start to disentangle them. To quantify behavior, we developed a setup that allows users to record and analyze the behavior of individual ants in many colonies in parallel. So the setup consists of an array of very simple webcams that monitor an array of petri dishes, each of which contains an ant colony. In each colony, the ants are marked with a unique color code that, right. Okay. Now, the film is a bit buggy, but I hope you get the sense for what the software allows us to do, which is to essentially follow individual ants as they move about in their colony. And the first thing that the ants do always is to form a nest by piling together their larvae in one spot. So in this case here, for example, the nest is down here. In this case, it's up there. And from this point on, at any moment, you typically find some of the workers in the nest where they care for the growing larvae, while other ants leave the nest to forage, look for food, or dispose of waste, or to explore. So even in this pretty simple and featureless environment, those ants really display a form of behavioral organization that revolves around who stays in the nest with the young and who leaves the nest to forage. And this is really the simple form of spatially organized division of labor that I will be focusing on today. So how do we quantify division of labor from the images, essentially? Well, again, division of labor captures the separation of tasks between different group members. And that typically implies two things. First, that there's some degree of behavioral variation between the different individuals. And second, that there's also individual level behavior consistency over time. In ant colonies, tasks are spatially segregated. Again, nursing happens only at the nest and foraging only happens away from the nest. And that means that individual behavioral roles can be meaningfully described in terms of individual spatial distribution. From automated tracking, we get the spatial distribution of each ant in each colony. And for each ant, we calculated the spread of that spatial distribution, which we use as a proxy for the tendency to perform tasks outside the nest. So in those examples, here, the ant at the bottom has a low spatial spread and spends almost all of its time at the nest with the larvae. The position of the larvae is shown in pink here, while the ant at the top, who is genetically identical and actually came from the same colony, spends much more time away from the nest, roaming in the rest of the petri dish. So using this approach, we first asked whether division of labor could emerge in response to increases in group size alone, and if so, how early this could happen. And specifically, we expected that in small groups, all ants might have to do everything, but that as colonies became larger, the individuals could maybe start to specialize in certain tasks. And this is a long-standing prediction theoretically, but one that had been difficult to test rigorously in social insects before, in part because it's difficult to manipulate group size independently from potentially confounding factors like demography. So to test this, we set up colonies of different sizes, starting with single ants, the smallest possible colony size, and going up to colonies of size 16, using ants of the exact same genotype and age. And we measured their behavior in terms of the behavioral trait I just described, so the spatial spread. And this is how individual behavior responded to increases in group size. So in this figure, each point is an individual ant, and each column is one colony. And if you just look at the distribution of this data, you might notice that it seems to fan out as colonies become larger, so that some behavioral phenotypes, like those ants here, that spend really most of their time at the nest, only seem to appear above a certain group size. And this resulted in an increase in behavioral variation within colonies as the colonies became larger. And we could show that this effect could not simply be explained by sampling effects, which is shown here by the fact that the data in black falls outside of the blue resampled confidence interval. So in other words, ants in large colonies really differentiate behaviorally more than ants in small colonies. Now, if division of labor increases with group size, we also expected behavior to become more consistent as colonies became larger. And to measure behavioral consistency, in each colony, we calculated how the behavior of the different ants relative to each other was correlated across successive days in the experiment using very simple rank correlations. And we found that this measure of behavioral consistency also increased with group size and actually became significantly different from zero, so from randomness, in groups that contain as little as 6 individuals. So as group size increases, the ants become more different from each other, and their behavioral role also become more stable from one day to the next, even though they all have the exact same genotype and age. And in summary, this means that small increases in group size and in group size alone can generate the plastic emergence of division of labor among a very small number of nearly identical individuals. Now, of course, this early work focused on extremely homogeneous groups in which we had experimentally removed any form of variation that we could remove. And so next we wondered how division of labor would operate in even slightly more complex social groups. And in this more recent work, we started introducing controlled amounts of heterogeneity by manipulating 3 different aspects of colony composition independently from each other. And so we manipulated genetic, demographic, and morphological composition. To measure the effect of colony genetic composition, we set up genetically pure colonies composed of a single clonal genotype called simply A and B here, as well as mixed colonies composed of a 50-50 mix of ants of the 2 genotypes. And as before, we quantified behavior in terms of spatial spread with higher values on the y-axis here, indicating a tendency to behave more like a forager than like a mouse. And we found that in aggregate, colonies composed of ants of one genotype forage more than colonies composed of ants of the other genotype. But that when those genotypes were mixed together in the same colony, then the behavioral differences between them became smaller, which is shown here by the fact that the blue and red dots get closer to each other. So mixing ants of different genotypes made them behaviorally more similar to each other than they had been in isolation, which is a form of behavioral convergence. We then used the same approach to manipulate colony demographic composition, this time by using old and young ants. And we find that in itself, age also affects behavior, so that on average, colonies composed of old ants, or it's more than colonies composed of young ants. But in that case, mixing ants of different ages had no detectable effect on their behavior, so that they behaved in the same way, irrespective of the demographic composition of the group. And finally, we manipulated colony morphological composition by using ants of slightly different sizes. And colonies made up of either large or small ants showed really modest, minimal differences in behavior that were actually not statistically significant. But when those individuals of different sizes were mixed together in the same colony, then they became behaviorally more different than they were in separation, which is a form of behavioral divergence. So basically, each source of heterogeneity in colony composition led to a very distinct outcome, which really came as a surprise to us at the time we did those experiments. And of course, we wondered how we could explain those observations. And as a starting point, we asked whether classic models of division of labor could explain our empirical results. And I'm calling them classic because they are quite popular in the field and they are often used to interpret empirical data in social insects. And we focused on one of the simplest and also one of the best studies models of division of labor, the so-called fixed threshold model of division of labor, which was first proposed over 20 years ago by Bonabo and colleagues. So this model assumes that individuals in a group differ in their response threshold, so their sensitivity to different stimuli that indicate the colony's needs. So here, for example, a stimulus that indicates how hungry the larvae are. If no one works, the larvae just get hungry and the stimulus increases in intensity until it hits the threshold of the most sensitive individuals in the group, in this case, the yellow ants. Those individuals respond by performing the corresponding task, in this case, foraging. And by doing so, they decrease the stimulus intensity. So task performance decreases stimulus intensity. And by performing this task, basically the yellow ants keep the stimulus intensity below the response threshold of the other ants in the group. And because they are consistently the first ones to respond to the stimulus, they end up specializing on that task. Now, if the yellow ants should disappear for any reason, then the stimulus can simply keep increasing in intensity until it hits the threshold of the orange ants, who can take over the foraging task. So the appeal of this model is that with those extremely simple behavioral rules, simple enough for ants, it can explain the emergence of a form of division of labor that is both robust in the sense that it can lead to individual specialization, but also flexible in the sense that it can adjust to perturbations. So to ask whether this particular model could explain our own empirical observation in ant colonies, we teamed up with Mari Kawakatsu, at the time a PhD student in the lab of Corina Panetta. And Mari simulated pure and mixed colonies as we had used in our experiments, assuming that the two types of individuals only differed in their mean response threshold. So here, for example, ants of type X have a lower threshold than ants of type Y. And the predictions of this basic model in terms of the performance of any given task were actually much simpler than the empirical observation. So in pure colonies, ants with different thresholds actually work the same amount because it takes the same amount of work to keep the stimulus intensity at their respective levels of tolerance. Then when they are mixed together, then the ants with the lower threshold, in this case, the red ants, end up working more than the ants with the higher threshold, the blue in blue, because they consistently respond first to the stimulus. So essentially, the model predicts that if individuals in a group differ only in response threshold, then they should always diverge behaviorally when they are mixed together. And clearly, this simple prediction could only partially explain what we had observed in real ant colonies. It could explain cases of behavioral divergence, but not cases of convergence or cases with no effect of mixing. And so this discrepancy prompted us to consider variation in other model parameters that we thought were biologically relevant. And one parameter that was a particularly good candidate is task performance efficiency, because ants in nature have often been described to vary in how efficiently they perform different tasks. In the model, task performance efficiency captures by how much ants decrease stimulus intensity by performing the corresponding task. And to make a long story short, we found that if we allowed the different types of individuals in the model to vary not only in their response threshold to a stimulus, but also in how efficient they were at decreasing that stimulus intensity, then the model could recapitulate the spectrum of empirical findings, at least qualitatively. And it was essentially the relative strength of variation in efficiency and variation in threshold between the two types of individuals in the model that determined which scenario the model produced. If variation in efficiency was high relative to variation in threshold, then the model produced behavioral convergence in mixed colonies. If, on the other hand, variation in efficiency was low relative to variation in threshold, then the model produced behavioral divergence in mixed colonies. And when those two forces balance each other out, then there was essentially no effect of mixing on the behavior of either type of individuals in the model. So in summary, for that part, we used the features of the clonal radar ants to quantify the relative importance of inherent individual traits and of the social environment in determining both individual level behavior and colony level patterns of division of labor. And we find that both the direction and the magnitude of those effects depended on the specific type of heterogeneity that we had experimentally introduced in those colonies. Theoretically, we find that on its own, a classic model based on threshold, response threshold could only partially recapitulate our empirical findings, but that introducing one other source of variation in that model allowed it to at least qualitatively capture the full spectrum of empirical findings. Of course, as a model, this still only provides one explanation for behavioral observation, but it helps us generate testable predictions that we can now go back to the lab and investigate. And perhaps more importantly, what I presented up to this point gave us a much better understanding of the factors that influence behavior in the clonal radar ants. And based on this, we can now start to look at what some of the consequences of that behavioral variation might be. And as I mentioned earlier, the research of my group is now being focused on how social organization might affect disease susceptibility. This is a newer research direction and one that will expand in the future as the lab grows, but this has, up to this point, already very much been a team effort by several past and current members of my group across now 3 institutions, and thanks to Camille from the European Research Council. So what's the motivation for this project? Well, we know that disease has very important impacts at different scales, 2 of which are individual infections and group level epidemics. But there's often a gap in the way we study and understand disease at those different scales. So on one hand, individual infections are often studied in quite some detail in classic model systems and under very well-controlled laboratory conditions. Epidemics, on the other hand, are of course more difficult to study experimentally, so we often use theoretical models or observational studies in naturally occurring epidemics. But at that scale here, we rarely have full knowledge and even less control over who is in the group in the first place. And this gap sometimes limits our understanding of how the properties of individual group members affect global level disease dynamics. And conversely, we don't fully understand how the likelihood for one particular individual in a social group to become sick depends on the properties of that individual social group. And one group property that is thought to be quite important in that respect is the structure of the interaction network, which represents with whom and how often individuals interact. So practically, the way we are approaching this question is to use experiments to study the causal link between group composition and infection dynamics. And as I mentioned earlier, we are particularly interested in the parts of those effects, if any, that are mediated by social organization, so by who does what in the group and who interacts with whom. We use the clonal radar ant to manipulate and replicate group composition along several axes that I mentioned earlier in this presentation. And we use automated tracking to measure how this affects social organization and in particular the structure of the interaction network. So this is somewhat of a technical point, but in order to capture all the interactions that occur between all group members, we need to work at much higher temporal resolution than in the data I presented earlier. And this provides much more detailed information about the spatial distribution of each ant in each colony. So in this data collected by a PhD student in the group, Giacomo, each black, you see the trajectories of 16 ants with identical genotype from the same colony over about 4 hours of recording. So each black circle here represents the spatial distribution of one ant. And you probably immediately notice that there's quite some variation in how those different individuals use their space. And this variation comes at least in part from the fact that in this particular colony, Giacomo has mixed old and young ants, which again turned out to differ in the propensity to stay in the nest with the larvae, which is what the young ants do, as opposed to roaming in the rest of the petri dish. From those individual trajectories, we can now build networks of pairwise physical contacts between all group members. So here the nodes represent individual ants color-coded by age, and those nodes are linked by edges, the thickness of which indicate the frequency of interactions. This is of course just one example network from one colony, but using those same tools, Giacomo and other group members have started measuring interaction networks in replicate colonies with different compositions, again using very simple manipulations of group composition to create homogeneous or heterogeneous social groups in order to identify networks that differ in their predicted epidemiological properties. So this is a form of behavioral screen, but not for individuals, for whole social groups. So this is still work in progress to some extent, but what we are asking now is first, well, how does group composition affect network structure? And second, we are also asking how disease is theoretically predicted to spread on those empirical networks according to different assumptions and theoretical models. This is one example in which a postdoc in the lab, Daniel, simulated disease spread using a very simple susceptible infected model in the temporal networks of our ant colonies over just 10 minutes of data. And you can see that based purely on the patterns of interactions, so based purely on network topology, disease spread expressed here in terms of the number of infected individuals over time is faster in colonies composed of young ants than in colonies composed of either old or mixed age cohorts. So in the next step, of course, we want to start testing some of those predictions experimentally. So we are now starting to inoculate our networks with different parasites and pathogens, and in order to measure how they propagate across those networks over time. And for this, we use, for example, one class of generalist entomopathogenic fungi, but also nematodes that are more specialized and naturally infect a specific part of our ant's head. And I want to discuss those nematodes in a little bit more detail, because even though there's still a lot we don't understand about them, what we know seems to indicate an interesting relationship to the social biology of their host. So those nematodes belong to a genus called Diploscapta that is actually a close relative of C. elegans, but that has repeatedly been reported in ants in the literature as early as the 1960s. The ones are pretty infectious in the sense that most ants in our lab colonies become infected a few days after eclosion and can carry anything from 1 to about 100 worms per ant. In this micro-CT scan produced by our collaborators in Japan, you can see how the worms in blue are really restricted to a specific zone in the head of our ants. The ants are shown in orange. And this zone is actually a gland called the postpharyngeal gland, which has many different proposed roles, 2 of which are larval feeding with potential downstream effects on cast fate, as well as in the storage and spread of cuticular hydrocarbons, which in social insects play an important role in social communication, including nestmate recognition. Now, many nematodes are phoretic on insects, in which case they typically just use their host for dispersion and don't necessarily have a negative impact on host health. And so the first thing we looked at is whether those nematodes actually do have negative consequences for the host. And it's clear here in that case that they can have large detrimental impact on host survival, as shown by decreased survival in experimentally infected colonies. We also see that infection risk is actually not equally distributed across the different group members, but it's instead really linked to the ants' behavioral goals. So Talita, a former master student in the lab, showed that in colonies in which all ants had the same genotype and age again, the individuals that acted as foragers, shown here in dark blue, became infected earlier and carried higher infection loads than the individuals that acted as nurses in light blue. Again, even though they all had the exact same genotype and age. And we are now, of course, exploring whether this pattern of infection is linked to patterns of interaction within and between those two different behavioral groups. We now want to use this and other parasites and pathogens to ask first how does the position of an individual in the network of its colony affect its likelihood of getting infected? And also, maybe more importantly, what are the specific properties of social groups that actually reduce disease spread? We also study the more proximate or the more immediate drivers of disease-relevant social behavior. For example, we know that collective behavioral defenses against disease, like the mutual grooming that I mentioned earlier, they often rely on the detection of sick or infectious individuals by their nestmates. But how this is achieved in practice is not always very clear. And this is an example using the entomopathogenic fungi that I mentioned earlier in experiments run by Yuko Ulrich, a former postdoc at the Berkeley University. So this fungus can be quite deadly. So here, all clonal raider ants exposed to spores of this fungus die within 9 days. And they are just exposed on their body surface in this case. But if after exposing the ants to the spores, you keep them in a group instead of alone, then the survival increases dramatically. And that's because immediately after exposure, the ants that have been exposed to those fungal spores receive really intense grooming from their nestmates, which essentially removes the spores from their body surface before the spores can germinate and penetrate the cuticle. So the ants are able to rapidly detect the presence of the pathogen, and they help their nestmates survive. And this is actually a very common phenomenon in social insects in general. But in this scenario, the ants are likely able to detect the pathogen directly because it's present in very large amounts on the body surface of the host. And it's clear that in nature, there are many cases, perhaps most cases, where the pathogens are not carried on the body surface and might not be that easy to detect directly. And so we wondered whether ants could use indirect cues, cues emanating from the host itself, to detect the presence of a pathogen in the colony, and specifically whether they could detect the immune status of their nestmates. And to test this, Jeremy, also a former student in the lab, used so-called immune challenges, in which he injected some ants in a group with beta-glucans, which mimic a fungal infection and induce a strong immune reaction, but are neither pathogenic nor contagious. And he found that immune challenges can induce the same qualitative behavioral response as an exposure to real pathogens, in the sense that challenge ants here in blue receive more grooming than their control injected nestmates in orange, even though the response is smaller in magnitude and comes in with a little bit of a time delay. But that means that ants indeed seem to be able to detect the immune status of their nestmates. All right, so I want to end this by giving a little bit of a summary of what I've discussed and try to put it in the broader context of the causes and consequences of behavioral plasticity and diversity in a social context. So we've seen how a number of individual traits can affect individual behavior independently from each other. Today I've mentioned only age, genotype, and morphology, and I've briefly touched upon how immune status can affect individual behavior. Then I discussed how the social environment can modulate those effects, focusing first on group size and then on group genetic, demographic, and morphological composition. With the example of the fungus infection, we also saw how just having a social environment at all, as opposed to being alone, can modulate infection outcomes via effects on behavior. Beyond behavior and infection, the social environment is also expected to have large effects on other important traits, such as longevity or reproduction, which I have not talked about today, but which we are also observing in many ongoing experiments. All those traits are likely affected not only by the social environment experience as an adult, but also by the social environment experience throughout development. for example, throughout larval development. And this is especially likely to be the case in social insects, where the larvae really rely on the workers for care and food, and where small differences in nutrition at critical stages in development can really lead to large morphological differences in adulthood. Then one thing that we are quite interested in at the moment is also to look at how those effects play out over time, and especially across generations. And because generation time is actually much smaller in the clonal variant than in other social insects, it's just 1 month as opposed to often 1 year. It makes it possible to study the effects of the social environment or of different experimental manipulation, such as immune challenges on behavior and immunity across generations, for example, through epigenetic effects. And finally, the ability to simultaneously observe all parts of the system, so all individuals in the group, over relatively long time periods, makes it possible to comprehensively study how the parts interact to produce group-level phenotypes, such as disease dynamics and evolution, and also various facets of group performance. For many of those longer-term questions, we can actually leverage the ongoing development of new tools for the clonal variants. So, for example, the continuous improvement of computational behavioral analysis now allows to study behavior at much higher resolution, but still in groups of freely behaving animals. For example, by observing how different body parts move in relation to each other in order to discriminate different types of individual behaviors, but also different types of social interactions. And the clonal variant is one of the rare ant species where we can now do basic genetics, including genome editing, which has now opened up the exciting possibility to use ants with altered behavior or immune phenotypes in experiments. Okay, so with this, I will end by thanking a large number of people across institutions and countries. For the first part of the work, Daniel Kronauer and my colleagues in his lab, as well as neighbors and collaborators at the Rockefeller universities and other institutions. And for the second part of the talk, all the past and current members of my team in Lausanne, Zurich, and now Jena. Thanks a lot for attending this, either online or in person. Here are some ways to find more information about either our research or to get in touch with me. And I'll be happy to take questions from either part of the audience.
So we will jump from question from here present the audience to online, if there's some. So we'll start from here. Any question in the room? Yes, Oli. Oh, sorry. I will repeat the question for the people online because they may not hear that.
So in many of your experiments, mix ants from different sources, phenotypes, or different types. Is this what's happening in natural conditions as well? Something mixed from, colonized from different sources?
So I rephrase. In the experiment, we mix genotypes, and the question is whether in nature this actually occurs as well, or whether there's just separate colonies just being separated.
Yes, so the short answer is for genetic mixing, no. And for the other two types, yes, it happens in nature. So age, of course, happens naturally, right? Because they reproduce every month, and then you end up with colonies that are naturally demographically diverse. Size variation also is natural, not in the ratio that we have used. It's more of a 95-5% type of variation. Genetic mixing is a weird one because it's true that in most ant species, you cannot make functional mixed colonies simply because the ants will fight to death. And in this species, it's not the case. So we can very easily mix ants of different genotypes, and they will form functional colonies that persist over time with no mortality to speak of. We don't really know why that is the case. I have a suspicion that the nematodes might have something to do with it, but I'm not quite sure.
We now have a question from Anne-Geneviève. Anne-Geneviève, you can ask the question.
You just need to activate your microphone.
Do you hear me? Can you hear me?
Yes.
Yes, we can hear you.
Okay, you was talking about a gland where you can find cuticular hydrocarbons used for recognition in ants.
Yes.
You were speaking about the postpharyngeal gland or another gland?
No, exactly that one, the postpharyngeal gland. Yes, that's where we find the worms.
Okay, okay. Ah, it's interesting. Okay, thank you.
Question in the room. Ali.
So you can remove the donkey by grooming. Does this not increase risk of infection for the groomers? Or does this become a special sort of task force which some do?
Just to get question.
Sorry. Can mutual grooming actually incur costs for the groomer in the sense that it could lead to their own contamination with spores? So the answer is yes, but the risk is probably extremely small. So when the ants groom, they actually ingest the spores and they pack them in their mouth, which in a part of the mouth called the infrabuccal pocket, and then they spit them back out. So it seems to essentially deactivate the spores. There is some amount of transfer of spores to the body surface, but this amount is typically too low to then induce death. So the idea is that yes, it kind of produces, it does result in the transmission of some spores, but not to the extent that it could actually kill the groomer.
Online questions? Then Manu, please.
So in the last part, you showed that these new challenged ants were recognized.
Mm-hmm.
Do you know how that works?
So do you think they could hear? So the question is, how could the recognition, the detection of the immune status work? I didn't mention that, but we looked at the usual suspect with ants is usually those cuticular hydrocarbons, which encode a lot of information. And we looked at that also in part because this has been checked in other systems upon following immune challenges. And in past work, people had found differences in cuticular hydrocarbon profiles. We did not in this particular case. It's one of those cases where I'm not quite sure whether it was a question of statistical power or something else. I'm not fully convinced that they do not play a role, but we could not find any effect in this particular work. It could, in principle, also be behavior itself. If it changes the behavior of the challenged individual to an extent that it can be picked up by other individuals, that could also be a way that nets could detect.
Because there must be a delay for the hydrocarbons, right?
Yes, but there was a delay in behavior as well. Yeah.
Yeah, I was a bit surprised that you use the spatial spread or variation in the spatial spread as a measurement of division of labor rather than variation in position itself, or structure that says that individuals occupy different places. Is it because they all have to go back to the nest anyway, or something? Or don't they actually occupy different places?
So the question is, why did you use spatial spread as a measure, indirect measure? Yes.
Instead of spatial position.
Yeah. Okay. So this is actually a practical reason. So basically, we do not force them to. The nest can be anywhere in the petri dish. With automated tracking, you don't locate the nest, so you just have the position of the ants. And so we were looking for a measure that kind of takes into account both the two-dimensional aspects of data, but also gives us a proxy for whether what we wanted to know is whether the ant is in the nest or outside. Right? But we don't know where the nest is based purely on tracking. So we use this as a proxy.
Yeah. So my question is: is the division of labor really reflected by how much they move, how they spread, or is it reflected by where they are? See what I mean? Regardless of where they're.
So the answer is division of labor is reflected by where they are in relation to the nest.
Okay.
Yes, because the tasks are spatially organized, of course, but very much around where the brood is. And yeah. So in many other experiments, people use inside, outside, right? So they would have a box where you're like in and out. You can't do this with those ants because they are blind and they are not sensitive to inside, outside based on light levels. And so we use this approach. It's an indirect approach, but what we were trying to measure is whether they are in the nest.
I see.
And we validated that by manually tracking the nest and so on. So there's a lot of validation that we had to do, but I haven't mentioned.
Thanks.
But this is a proxy, really. how do the ants decide where they establish their nests? We really don't know. So when we set up those experiments, initially we wanted to determine the nest position ourselves by giving them a little cavity or basically trying to make them have the nest in the center of the box. It never worked. They would just put the nest wherever they wanted, and we could never figure out why they would put it in one specific spot, essentially. It's an interesting aggregation process, right? We give them larvae that are scattered. The ants have, within a few minutes, they essentially have piled up their larvae. How they decide where to pile it up, we don't really know.
I just curiosity question. I was wondering, like, once they choose a particular task to do, does that affect the likelihood that they will benefit from the grooming behavior or a particular diet that makes them more or less susceptible to get infected?
So the question is the amount of
So is the labor task,
yes,
they are within or
Mhm.
nest, does it affect the likelihood of being helped by others in terms of grooming, or whether they will access resources or more or less resources that would.
right? So I think the likelihood, I don't know. I can mostly speculate. So I think the likelihood of being groomed is probably more dependent, based on literature, not on our own work, based on simply who you encounter, right? So if you're out of the nests in a place where you're unlikely to encounter other ants, you're very unlikely also to receive any grooming. But if you are in a place with reasonable chances of encounter, then you will receive it. There might be some things modulating the likelihood to groom, for example, how valuable you are to the colony or how old you are, but this is not something we haven't looked at. When it comes to resources, usually the notion is that resources are shared within ant colonies, so like foragers bring back food to the nest, so in principle at least, the resources is similar in quality and quantity, I would guess, but again, that's pretty speculative on my part. Sorry, can you repeat the beginning of the question? Oh, do the ants crawl on the sea? No, so that would really mess up our analysis, and we are lucky that they don't do that because they are poor climbers, simply. Most ants are good climbers. That would be an issue. If they did that, we have to find a way to prevent them from doing that, because obviously you can't track them when they're upside down.
I have a question.
Yes.
So, well, maybe more a perspective, but so you expect an infection to rearrange or change the social structure, the network of the colony. So just on a simple one generation level, would you expect them, if the next generation would face the same challenge, if the social network would just respond as quickly, as faster as, do you have any idea about that? Whether this
across generations.
Yeah,
that's a really interesting question, right? So, well, in practice in ant colonies, when the second generation occurs, the generation overlap.
Yeah.
So, from the individuals that were present in the first round, you would expect perhaps faster responses through effects on experience. So this is known to occur in ants. You're faster the second time around. Whether this could be due to epigenetic effects, for example, occurring.
Yeah, I was thinking because you have this like strictly clonal generation, right? So where they double, and so you can have like the offspring as the parent, but just the parent experience the parasite and potentially arrangement of the network.
Yes, so basically we don't know. That's a very interesting question. I think so. Transgenerational effects are known via the immune system, of course, but on behavior, this is, to my knowledge, at least much less well described.
Yeah.
And that would certainly be something worth investigating, I think, yes.
Yes.
So in your introduction, you alluded to this network structure within a colony that might be optimal to slow down disease spread, right? And so I'm not an ant person, but from what I remember, not in your system, but in systems where you have queens, there's a lot of top-down control. so that not everyone starts to reproduce randomly. And I could imagine that a structure that would prevent disease spread would be different from a structure that would facilitate control. Is there any evidence that this somehow balances to form certain structures that allow for control but prevent disease spread, or is there what wins in that trade-off?
Okay. Can I try to reform it? Is the question like of a trade-off between information flow and disease?
It could be information flow. I just framed it negatively in control, but it could be information flow versus disease. Yeah.
So I think that's a really good question, and that's really at the core of the field, in the sense that often we study networks, but we don't really know what those networks convey, right?
Yeah.
It's not necessarily the same type of behaviors that will result in disease transmission or information flow. And I think currently the field is very limited in the sense that it's still looking at very rough coarse contacts, right? And as a proxy for things that might transfer either information or disease. But in reality, what's very likely to happen is that different type, you could construct many different network, social networks based on specific social behaviors, right? So for example, a grooming network might be a network that leads to the propagation of spores, whereas intonation network might be one that leads to the transmission of information. And those are, of course, now we look at bidirectional networks, but in reality, they are, information flow sometimes just one direction, and disease also. And so I think where we have to go from here is basically towards a more refined picture where we can start to discriminate between different networks that propagate different things in a colony.
Yes.
Is there a separation between these acts to the grooming world whenever the spores are on some act?
Yes. So the question is there specialization in grooming in colonies? It's been reported in some social insects, I believe in honeybees, and in our system we don't know yet, but we are actively looking at it. So I have a PhD student who is trying to quantify that. And we are particularly interested in this because they are clonal and have no other form of variation. So we would like to see whether some individuals become specialized in the sanitary tasks in an otherwise clonal colony. Yes.
And does the fungal spores affect the eggs?
Do the worms transmit via the eggs? No. To our knowledge, those worms do not infect any of the brood developmental stages. They only infect adults. So we can actually create clean colonies by removing very young adults.
I have a last boring question. How do nematodes transmit from the head? And is by direct contact?
So yeah, that's a very good question. And basically we don't know. So one possibility is that, so the contents of they sit in a gland that is connected to the mouth.
Okay.
And the contents of that gland are known to be transmitted between ants via grooming, for example. So that could be one option. In many ant species, there's trophallaxis, which is the mouth-to-mouth exchange of liquid food. Our ants do not have that. So it's possible in other ants, not in our species. But in reality, we don't really know yet whether that's the case. When we make colonies of infected and non-infected individuals, it takes a very long time before we see any transmission even. So if it happens by a direct contact, it's not a very rapid process. It might be that they get infected via the environment.
And this is also about the nematodes. In natural populations, you find that most colonies are infected.
Okay, so the question is what does the prevalence look like in natural colonies? For the clonal variant, we don't know because our colonies were, those are lab lines that have been propagated for like over a decade. And when we found the nematodes, they had already, they were in all colonies, so we don't know where they come from. In natural, so we also sampled other ant species to just figure out whether the nematodes were a real thing, and if so, how frequent they were. So you typically find within a species infected and non-infected colonies, and then you can also infect ants from one species with nematodes of sample from the other species. So there doesn't seem to be a high degree of specificity in the system. It's just very organ specific somehow.
Are there any online questions? Other questions in the room? Okay, then we can finish here. Thank you very much, Yuko, for a very super interesting talk. And if someone wants to talk to Yuko, she'll be here all afternoon. Building 22, second floor. Just come there or write an email, yes? Sure. And if someone wants to join as well this evening for drink dinner, just send an email or contact me and we can arrange that. From the technical side. Okay, so thank you again, everyone.